Okay, good morning, everyone, and welcome to the 2013 Royal Tyrrell Museum Speaker Series. Today, the Royal Tyrrell Museum and its cooperating society are proud to present Dr. Alexander Wolfe. Alex is a professor of paleobiology in the Department of Earth and Atmospheric Sciences at the University of Alberta. Alex is originally from Montreal. He obtained his bachelor's degree in biology at the University of Western Ontario. He subsequently moved to Kingston to pursue a PhD in the Department of Biology at Queen's University. For his dissertation, he studied lake deposits in order to reconstruct late Quaternary environmental change in Baffin Island in the Canadian Arctic. Upon completing his PhD, Alex held a postdoctoral position at the University of Montreal before moving to Boulder, Colorado, to hold a research position at the Institute of Arctic and Alpine Research of the University of Colorado. Then in 2001, he was hired as a professor in the Department of Earth and Atmospheric Sciences at the University of Alberta. In the years since then, Alex has also gone to teach at the University Center in Svalbard, Norway, and at the Université de Franche-Comté in France. Alex's research interests focus on the study of environmental changes over short and long time periods. Most recently, he has been involved in defining the Anthropocene, the era of humankind, using sedimentary records. Today, Alex will discuss the exquisitely preserved fossil plants found in kimberlites that formed 53 million years ago and their implication for models of future climate change. So, without further delay, I present you Dr. Alex Wolfe.
Thank you. Am I coming through okay now? I usually project pretty well. I don't want to be over magnified. So thanks to the museum and thanks for having me here. The objective today is to really synthesize some of the work I've been doing over the last 5 or 6 years on fossils that are associated with a rather unconventional stratigraphic context, and this is the Kimberlite magmatic system. And the Kimberlite, of course, is a volcanic edifice which originates from the mantle and brings things such as diamonds to the surface with these very, very typically what we infer to have been explosive phreatomagmatic events, and that actually, perhaps surprisingly, are associated with quite remarkable fossil deposits. So this is the story that I'll give today, and it comes in several parts. And I'd also like to additionally discuss some of the implications that our observations on the paleoclimates that we're able to infer from these deposits, what some of these inferences might be for with respect to climate futures on our planet. So what I'll discuss first is the Kimberlite system as a host for fossils, and here we're discussing fossils of Eocene age. So there are two deposits. One is approximately 40 million years. The other is 53 million years, and the Eocene as a whole is generally viewed to be the time between about 55 and 33 million years before present. I'm going to discuss some of the paleobiology, as was said in the introduction, with an emphasis on higher plants, but also on some microbial components, and I'll discuss a little bit of palaeontology and some geochemistry as I go along. I've been working quite a bit on trying to understand the geochemical message of some of these materials, with a particular emphasis on stable isotopes, and I said before, some of the implications for the future. Now, if you're going to doze off, the message is that the Kimberlite system, despite being a violent form of magmatism, can offer the potential in at least two distinct geological contexts, one being within the Kimberlite body itself, whereby material tumbles into the magmatic rock and becomes entombed, and the second being the post-eruptive infill of the Kimberlite pipe, which is this carrot-shaped thing intruding the country rock, that both of these geological contexts offer the potential for truly outstanding fossil preservation in a region that, because of the effect of combined Phanerozoic land denudation and pervasive glacial erosion during the Quaternary, offers very, very little else to interrogate from a paleobiological perspective. So, well, I keep going the wrong way, and I'm going to miss one if I'm not careful. So, of course, this work isn't mine and mine alone, and I've been really privileged to have some outstanding collaborators with which to embark on this journey. And first and foremost, they're Carlos Muhlenbach, my senior colleague at the University of Alberta, who is an extremely well-known isotope geochemist who's worked on everything from fingerprinting fugitive gases coming from leaky wells to the origin of life and everything in between, including peridotites in the mantle and meteorites, and a former PhD student of mine who will be here giving a lecture in the near future, Ryan McKellar, who's also been instrumental. And so this is our local team. And then we have two alumni, Ralph Tappert, who's now at the University of Innsbruck in Austria, and Alberto Reyes, who's now at Queen's University in Belfast. When they were students at the University of Alberta, were involved in developing our database. And then we have the Connecticut connection, Peter Siver, who's a botanist at Connecticut College, Dana Royer at Wesleyan, and Gabriela Doria, who's now doing a PhD in the forestry department at Yale, have been instrumental in putting together various parts of the data set, particularly Royer and Doria, where it comes to estimating the ancient levels of carbon dioxide in the atmosphere from leaf stomatal indices, and others, a cast of people. Some will be known to members of the audience here. John Westgate, who is an expert on volcanic deposits, Art Sweet, a paleontologist. David Greenwood down the road at Brandon has been involved in statistically converting paleobotanical data into paleoclimate data. And Adam Sank, who's now a professor at Nipissing, is a paleobotanist who's been involved in some of the chemical extraction and purification of the materials that I will discuss. I'm going to keep going one back and two forward for the rest of the talk, I can tell. That's just the way it's going to be. Oh, no. Okay, so roll towards me. So what is a kimberlite? So the kimberlite is named, of course, after the type locality, which is Kimberley in South Africa. These are volcanic systems that originate at considerable depth, typically in the range of 100 to 120, 130 kilometers beneath the surface. They are typically highly volatile. They contain considerable amounts of gas, mostly carbon dioxide under tremendous pressure, and quite a bit of water. And they ascend episodically in Earth history to reach the surface, where they explode catastrophically in events that we call phreatomagmatic. And so the magmatic interaction with groundwater and highly pressurized fluids, leading to these astonishing eruptions that leave holes behind that then become infilled with the mantle material that then delivers with it materials from the mantle, including well, diamond is a big one, but also clinopyroxenes and pyrobes and other mineral species from great depths. And kimberlites are only known in the modern landscape from stable cratonic settings, and that's what we see right here. And so this might be the heart of Brazil, or of North America, or of Greenland, or of South Africa, these stable cratonic settings which then preserve the intrusion without significant faulting or metamorphic alteration after the intrusion. So I'm not going to lecture much about the kimberlite per se, because I'm not a volcanologist, but the volcanological underpinnings are nonetheless important in sort of generating a holistic perspective of the system when we aim to interrogate it from a paleobiological perspective. So this is a very cute picture from De Beers. It's a depiction of the late Cretaceous Orapa kimberlitic intrusion in Botswana, which has been mined. And so this is the kimberlite hole. So imagine that carrot-shaped thing that we call the diatreme going down to considerable depth. And then after the dust has settled, a few theropods, some sort of eutherian grade mammal eating, I believe, is an odonate or something. It's eating, no, it's maybe a beetle. not an audinate, that's a piece of the branch. Various flora and fauna representing the Cretaceous environment, a few amphibians, and of course a big stone right here, which is the main purpose, of course, to understand and exploit these regions. And so over the years, over the decades, there have been an assortment of reports of fossils associated with kimberlite, and that forms the basis. I'm just providing one of the first reports from the Canadian kimberlite field, which is the Canadian kimberlites were only discovered in the mid-'80s. But we've known for decades that in places like Orapa, which is late Cretaceous, and from my perspective, more spectacularly at Mahenge in Tanzania, where the post-eruptive shales contain the earliest cichlid fishes and other things like cynodont catfishes, some of these organisms that have then radiated tremendously in the African Rift Valley lakes. And there's also a really nice flora of magnolia vegetation from this site. We have known from scattered occurrences from the African continent that kimberlites can be kimberlites can be associated with fossils, and fossils of considerable quality. The difference between Canada, of course, and South Africa is the legacy of the Laurentide ice sheet. And so in Canada, we don't have perfectly round diatreme holes sitting on the landscape because the landscape has been so pervasively denuded by glaciation. So we see some of the primary structure in the Canadian Shield, a lot of muskeg and modern glacial lakes. We don't see anything on the modern landscape that resembles what we see in southern Africa. And of course, the discovery of kimberlites on the Canadian soil was not done the way it was done in Africa by screening alluvium and so on. It was done by the combination of aeromagnetic surveys to identify the pipes that have no visible surface expression in combination with sampling of discrete surface sediments or glacial sediments, and screening these sediments and looking for microdiamonds and clinopyroxenes and other mantle-borne mineral species. And then when the concentration of these things is the highest, presumably you're close to the pipe, and then eventually you zero in. And companies, not Canadian companies, unfortunately. Well, no, some Canadian companies, but in particular giants such as De Beers and BHP, Broken Hills Property, an Australian multinational who've invested considerably, they have now collectively have documented in the range of thousands of kimberlite pipes on the Canadian Shield. And we're specifically interested in this region right here of the Northwest Territories, sort of halfway between Yellowknife and the Coronation Gulf, which is up here in the Arctic Ocean, a region called the Lac de Gras kimberlite field, which has hundreds of pipes that range in age from Eocene to well into the Paleozoic, and some of them are indeed reveal economic grades of diamonds and have been exploited. And so BHP's property, which has now been sold to Harry Winston, which has sort of indirect implications for the future of this kind of work, not only did they document many dozens of kimberlite pipes, but they also drilled in excess of 10 kilometers of exploration core before the recent sale of the property. And we were fortunate enough to be able to retrieve some of this core, in particular core facies that have nothing to do with kimberlite itself or to the diamond trade, but are rather of interest for paleontological purposes. So this is where we are in this part of the world, and several of these have been mined, both at Diavik and the Akati mine property, which involves 3 kimberlite pipes, and there's another one called Misery, which is also being mined at present. So what do these rocks look like? This is typically, I don't know, and again, you know, I'm not a volcanologist, I'm not an igneous petrologist either. This is a darkish, more magmatic sort of pure composition, and you sometimes see this. This is the stuff that really comes straight from the mantle. This is a little bit more admixed with other materials, including xenoliths and other scraps that have been incorporated on the way up. And this is what we'd call, and so these are what we'd call volcaniclastic kimberlite. And this in particular, this beautiful specimen, is what we'd call the reworked volcaniclastic kimberlite, because as it turns out, after these eruptions, the kimberlite settles and swashes in the diatreme before ultimately settling, and you can indeed see structures of imbrications, and you can even see, in some examples, you can see primary sedimentary structures in the kimberlite, which is quite curious to see, like wave ripples in a rock that has come from the mantle. And it's really quite remarkable. So this is what I would call a brecciated volcaniclastic kimberlite. And that's indeed from the Panda mine. So this is essentially the ore, and these are, of course, core samples. And so xenolithic material that shouldn't be there, and indeed isn't there when the kimberlite is pure. So this, of course, is an olivine. I think this is pyrope, but don't quote me on that. You can quote me on olivine here. This is ground olivine kimberlite mass. And then, of course, you get stuff that is collapsed in during the event, because the intrusive event is very, very violent. There's lots of water. There's lots of shattering of the wall rock. There's collapse into the hole, and you get things that are really quite interesting. Well, this is granitoid. This is a country rock. It's not that interesting, but this is a section where, we're trying to core through kimberlite to assay the diamond grade, and whoa, 30 meters of granite. That's quite common. This is a raft of granite that collapsed into the diatreme, and then when you take your core, you just end up coring through the granite. That's 30 meters of core at $300 a meter, where you're not going to find diamond because it's granite. And then at other times, what else can we have for xenoliths? This here is wood, and so is this twig right here. And this is wood that has collapsed into the kimberlite at the time of the kimberlite intrusion. The only mechanism I can imagine is that the kimberlite explodes. So you have the phreatomagmatic event that forms the diatreme, and the diatreme is slightly underfit, or the blast zone is slightly underfit to the diameter of the diatreme, and then wood, trees that were living at the site happily one moment, collapse into the hole a moment later, and most of the wood, of course, would be instantaneously pyrolyzed and blown to kingdom come, but some bits are actually able to enter the kimberlite body, presumably in a narrow boundary zone adjacent to the blast zone, and indeed can be retrieved at depths of several hundreds of meters beneath the surface. And so talk about finding wood or finding fossils in places you wouldn't intuitively expect, and this is it. And yet we see it again and again. These are unpermineralized stringers of small wood fragments, and so you can take this stuff out and you can crunch it up, you can even burn it, and just to show that this system actually works the way I'm claiming it does, this is a piece of this wood with pressed into it, occluded to it, a microdiamond. And so what I say in this room stays in this room. I didn't actually give this stone back to BHP. I thought, finders keepers. It's actually only 200 microns long, so it's not worth anything. But it's amazing, really, to me. So this is the cellular structure of wood that is contemporary with the moment of intrusion, which in this case is 53 million years before present, to which has been adpressed a diamond, which is probably in the range of 2.5 to 3 billion years in age from the Archean of the Earth. And this whole system is integrated together in a ground mass of tough acidic kimberlite. So this is the kimberlite that's really, really homogeneous. And I actually told one of my petrology friends, says, kimberlite is homogeneously heterogeneous. And he actually agreed with me. He said, oh yeah, you're right. And the man had a heavy British accent. It was all very funny because I don't know anything about kimberlite. He thought, that's really insightful. And so who knows? So so it always, I think it's useful sometimes to get involved in things that you don't know anything about. And so the two depositional contexts that I'm going to describe to you today are the following. One is entombment at the time of emplacement of the kimberlite, and the second is the post-eruptive sedimentation, and this is what we call Mar sedimentation. And this is actually a model that is quite well documented from volcanic crater lakes worldwide, including the Eiffel and Crater Lake, Oregon, and so on and so forth. The first one is a little bit more not subtle, but not predicted per se. And this is where the kimberlite then coming from the mantle. So this is the diatreme. And this is the narrow zone, presumably, that must have existed, that I'm not quite sure what the conditions were in terms of temperature and pressure, but a zone where vegetation can enter the blast zone and then collapse to considerable depths. When I say considerable depths, this is in the range of hundreds of meters here. So this is half a kilometer down. Sometimes the necks of these kimberlites right at the bottom are only but a few meters across, and the typical diameter in the slave province is a couple of 100 meters across. So actually they're quite a bit smaller than the African counterparts. But anyway, we get this rock mass as the one that I've just shown in the previous slide, which incorporates wood, xenolithic material, whether it's wall rock or any other business, either falling down or entrained on the way up. So that's the homogeneously heterogeneous part. And then when all the dust settles, in certain cases where the country rock isn't fractured, enabling drainage, a sedimentary basin can occur, and then you have this nice quiescent post-eruptive sediment. So here a little bit of lake sediments. Here the lake has, the lake basin has become infilled with sediments, and so you have a forest floor with renewed growth of trees and more of a peat-like sediment, and this is exactly what we have in the Giraffe locality. And then glaciation that backfills the hole and does a little bit of surface erosion, and then leaves us the landscape that we see today. So they're hidden fossils, they're cryptic fossils, but they nonetheless exist, and they exist in an area that offers scant alternate sediments of contemporary age with fossils of this age, and this can be said for a very, very large area of the Slave Province, which is essentially just muskeg, Canadian shield rocks as far as the eye can see. So that's my, so it only took me half an hour to do my introduction. No, we started a bit late and so on. And so I'm just going to go through results from the two contexts that I've shown here and tell you what we've found. And so of course, I'm totally dependent on the recovery from the diamond companies and for them to tell me that, this core is not worth anything for us because we can't do anything with respect to diamonds. You can have it for your paleobotanical business. Just put, the name of the company and the acknowledgments and so on. And we've been happy to do this with BHP and haven't had any success with any of the other companies. So this is the BHP mine at Ikati with 3 pipes, Panda, Koala, and Koala North. So this is viewed from the north, viewed from the south, looking up this way. This is what it looks like in plan view. So these are small kimberlites, and this is what Panda looks like, and wood is quite common in the kimberlite body that has been mined out. Well, now the whole thing just about has been mined out, and I'm going to report on a specimen at about the base of the wood-rich zone from 315 meters down in the kimberlite body. The kimberlite itself can be dated quite well, and of course the assumption here is that the wood accumulated in the kimberlite at the time the kimberlite exploded. And so if we take phlogopite, which is a phyllosilicate, a magmatic phyllosilicate from within the kimberlite, which is this ultramafic olivine-rich rock body, and we do the rubidium-strontium chronology, yielding a really well-behaved isochron, and this isochron is 53.3 million years before present, which is Ipresian, or early Eocene. The wood from the 315 meter bench looks something like this. So this is what it looks like in its native state, encrusted with volcaniclastic kimberlite. And this is a nice split face, which yields right in the middle here a pocket of amber, or conifer resin, that is polymerized. And I'm not going to talk about amber because there will be another talk in this series, but it is one of my sort of obsessions, and one of the motivations to work on this specimen in particular was because we could assay the quality of the wood, both morphologically and geochemically, alongside with that of the amber. So you really, despite being in this peculiar setting, 315 meters down in a volcano, we nonetheless have this sort of preservation, and this is unpermineralized, okay? So this has not been silicified or phosphatized or turned into rock. This is wood, and I forgot to bring the specimen, but I once gave this lecture, and I took a specimen and lit it on fire, because you can do that, and then I got in trouble from the university fire marshal because it's so bureaucratic, and it would probably be worse in the museum, so maybe it's just as well. But this stuff does burn. It is absolutely unpermineralized. It is exquisitely preserved. So this is the light microscopy of tangential and radial views of the wood anatomy, a science that we call xylotomy. These are rings of early and late wood. This is within rings. We have small crystals of calcite, which are probably following crystals of oxalate as pseudomorphs, and oxalate is a crystal that is naturally formed by plant metabolism. And then here we have the various components of the wood architecture, and so the canals, the rays and the canals and the border pits, and we have the whole arrangement, which enable us to taxonomically diagnose this as metasequoia, which is Don Redwood, a cupressoid conifer that now at present only lives in China. Sorry, I keep doing that. I'm really sorry. Maybe I should give this to Francois or something. So we now know that this tree from the xylotomy is this one, Metasequoia, or Don Redwood, which is a lovely, fast-growing, deciduous conifer. At present, it is only lives relictually in a few small mountainside copses in Hunan and Sichuan in China. But we do know that in the Paleogene, in the Eocene and Paleocene, and perhaps in the latest Cretaceous, this is one of the most abundant trees at high northern latitudes, where it has a panarctic distribution, for example, in the Canadian high Arctic, in Spitsbergen and Greenland, and elsewhere. And it's a real biogeographical mystery how this plant then became relegated to Southeastern Asia. I have some ideas, but it's a little bit outside the context of today's talk, and that it became biogeographically repatriated to Asia, all the more because in cultivar, this tree is actually quite easy to grow as long as you have enough water and your winters aren't too cold. And so the fossil record speaks very clearly, and there are similar, we're actually looking at similar fossils this morning from the late Cretaceous of this part of the world, and of course there are some taxonomic vicissitudes, but Metasequoia is, does have this opposite habit in the cones and the foliage, where you have one-to-one symmetrical matching, and that's a real diagnostic feature. And the xylotomy is pretty diagnostic as well. So what can you do with this wood, in addition to the xylotomy and the reconstruction that, of course, this tree was present, and of course, all the specimens, or about 90% of the specimens that we've looked at in terms of making thin sections and looking at them under the electron microscope, belong to Metasequoia or closely related cupressaceous conifers. We just have a few specimens of pinaceous trees from the Panda Kimberlite. But I haven't worked those up in this way, which is the extraction of the cellulosic material, which is the carbohydrate, the primary carbohydrate produced by the plant in a structural sense, and so it's a structural polysaccharide, and you have to use all these dangerous chemicals, and the most dangerous one is this one, where you're essentially making nitroglycerin, and so this is the part where Adam Sank did this part when he was a postdoc at the University of Arizona, and the rest of it we can handle at the University of Alberta. My colleague Carlos Muhlenbach actually has a terrible history with 2 fires in his labs and so on, so we have to be somewhat careful. But essentially, we're delignifying and removing any mineral precipitates with, with this strong reductant, which is it is hydroxylamine, hydrochloride, and then some base, and then more acids. And then what we end up with is a material that looks like this, just like you saw in my initial slide. And if you were to extract the cellulose from, say, I don't know, a 2x4 that hadn't been heat treated, and you're to do the same protocol on a modern, say, I don't know, the closest modern thing around here is cedar or juniper, you would get something that looked morphologically quite like this. And then when we do the middle infrared spectroscopy with the Fourier transform infrared machine, we then obtain from the Panda Kimberlite early Eocene wood that's 53 million years old, we obtain the same peaks for methyl and methylene in our extract that you see in the alpha cellulose standard from the chemical company, which we use to calibrate the signature. So this is just the absorption of individual infrared bands that are controlled by the positions of the carbon and hydrogen, and in some cases the carbon, oxygen, and the hydroxyl groups within the matrix, within this stuff, which looks like, visually, it just looks like white powder. And so we know we have cellulose, and in fact, this is the oldest cellulose, conclusively proven cellulose from anywhere in the fossil record. And that is because cellulose, there's a reason you can go to your shopping market, and if you don't want the plastic bags, you can get the cellulose bags, because the cellulose does degrade. It's tough stuff when it's in plant tissues and the plant is growing, but cellulose is ultimately, because it's a polysaccharide, it's just a complex sugar, it actually doesn't persist that well in the geological record, and yet here we have it 53 million years before present. Now, I don't want to bore people too much with the stable isotopes, because I didn't really know whether it was going to be a hardcore paleontology, geochemistry audience or not. And I see there's a lot of people from the sort of broader Drumheller community who are not necessarily geochemists. But what we can do with this material then, knowing that it's geochemically stable, is measure the ratios of regular hydrogen to the stable and rarer stable isotope hydrogen 2, which is deuterium, and do the same thing with the oxygen 18 to oxygen 16 ratios. And we do this, and we get values that are typically quite light, quite isotopically depleted, which makes sense because we're pretty far north. They're extremely narrowly constrained, and they're entirely reproducible. And it's actually a pleasure to measure this stuff isotopically on the isotope ratio mass spectrometer in Carlos Muhlenbach's lab. We can then use linear fractionation factors. So this is the difference between the cellulose and the water which the plant accessed to make the cellulose. So these factors relate to the metabolism of the plant itself, the biochemistry of incorporation of hydrogen and oxygen into the polysaccharide when the plant is living. And so these are established for modern plants, but we assume that they're equivalent for the fossil plants. And so we can then back out the inferred O18 and delta deuterium of the environmental waters that the plant was using. And this presents us with what we call an isoscape, or an isotopic landscape, which matches, I don't know, something like north central Saskatchewan or Manitoba, you know, the kind of places where you go pike fishing in the summer, but nothing like the subarctic. So it's it's clearly warmer, but it's not Florida either. It's definitely boreal, a boreal landscape when you look at these values. This one right here, no, sorry, this one right here and this one right here, and you plot them into modern isotopic space. They are quite, the hydrogen and the oxygen are entirely coherent. And then we can take these values and use different relationships developed for the Eocene and then do a whole bunch of statistical gymnastics. Some of it I like, some of it I don't like. Actually, most of it I don't like. And then you can derive what the temperature might have been at the time, and these end up with solutions of somewhere in the 7 to 12 degrees Celsius mean annual, which is over 25 degrees warmer than the mean annual temperature at Yellowknife today, which is about -5, and certainly warmest months that are indeed warm, but not tropical warm. And so this is looking pretty good. And so we've been thinking about these results, but ultimately there are easier ways to understand. And so if the idea is that the plant was living 53 million years ago, but it produced cellulose just like modern trees do, and this cellulose is faithfully preserved, and from the cellulose we can back out the composition of the water, how does it look by comparison to modern waters in precipitation at the closest place that these things have been measured, which ends up being Yellowknife. And so there's something called the Global Networks of Isotopes in Precipitation. It's been orchestrated for only a few years when it was funded by my colleague Tom Edwards at the University of Waterloo. And they have, I don't know, in the range of, I think it's 150 or so measurements of the isotopes of oxygen and hydrogen from surface waters in precipitation from the Yellowknife station. And we can see that our values, and so the GMWL, this is the global meteoric waterline. So this is the meteoric waterline that's predicated solely by the thermodynamics, the relationship of oxygen and hydrogen isotopic fractionation between air and water. And then the dashed line is the local meteoric water line, which is actually fitted to the to the available data from Yellowknife. And it diverges slightly, and that is due to the local effects of evaporation, which preferentially removes light hydrogen and light oxygen. It's a very slight effect because evaporation in Yellowknife, though present, isn't a dominant feature. And we look at our data from Panda 53 million years ago, and it fits on the isotopic space of the modern precipitation at Yellowknife, which is suggesting that, I'm not trying to say that the Middle Eocene in Yellowknife in the Northwest Territories was the same as today. It wasn't. There's no metasequoia there today. But I am saying that the general physical behavior of the climate system, the physics of it, was the same. And it was also within the bounds of the isotopic space defined by modern measurements, or at least modern measurements in the 85 to 89 interval, which is the sole one for which we have modern data. So if we just zero in on this box and look at it more closely, and instead of plotting all the values, just the monthly averages, so this is the mean of the 4 Panda measurements, and this is September with a mean temperature of 6 degrees, May, and then here June, August, and July, the warmest months in Yellowknife that are around between 13 and 16 degrees Celsius. And so they plot very, very closely to our Panda wood. And indeed, the Panda wood here, it's a bulk sample, or it's 4 bulk samples. I don't want to tell you how long it takes to purify the cellulose and measure it, but these samples are presumably annual integrations. But it looks like, on the whole, that Panda wood was metabolized under ambient temperatures somewhere in this range, which is about 20 degrees or so warmer than the mean annual temperature at Yellowknife today. And so the isotopes, you couldn't make up data that look better or more interpretable in terms of them being able to fit within what we understand of the modern geochemistry of stable isotopes, light stable isotopes in precipitation. So just the same data. Some people don't like co-isotopic plots of the hydrogen versus the deuterium, the oxygen versus the hydrogen. So here is just the temperature field and the hydrogen, temperature, and oxygen stable isotopes from the modern samples from Yellowknife, and then this is just the values from the panda hydrogen and the panda oxygen, and again, you can extrapolate them and they end up with temperatures somewhere between 10 and 15 or so degrees, probably a little bit on the warm side, particularly for the oxygen, maybe because that's reflecting only the period of active biomass accrual, which occurs in the summer. Remember, this metasequoia tree is deciduous. It's only metabolically active in the summer, and it sheds its needles in the winter, like a larch. Okay, so that's part one. There's only 7 parts to the talk. So yeah, I mean, I'm pretty excited because we have the oldest wood that has not only this beautiful unpermineralized anatomical preservation, which isn't completely unique. There are sites, for example, the fossil forest on Axel Heiberg Island, where there's unpermineralized pristine preservation. It's just that, you know, I know because I've tried, and it's not a competition anyway, but that stuff doesn't yield pure cellulose. It yields junk with a lot of iron on it. So we have the oldest demonstrable alpha cellulose preserved to date, and this despite the violence and the character of the depositional environment that this wood is associated with. And the climate was presumably much warmer 53 million years ago before present relative to today. We also know that because metasequoia needs at least 1,000 millimeters, or a meter, better yet, 1.5 meters of annual precipitation in order to survive. It's a tree that really likes to keep its roots wet, and the precipitation today in Yellowknife is around 200 millimeters, or 1/4 or 1/5 of that. We also know that the environment was much greater during the Middle Eocene. The environment was much wetter during the Middle Eocene than it is today. So a warm and wet early Eocene, that in and of itself has been demonstrated by some of the marine proxies and so on, but we can go several steps further in extending this to this region that doesn't have any other fossil deposits, and moreover, by interrogating the isotopic composition with respect to hydrogen and oxygen of the pristine alpha cellulose that we have from this hole, we're able to say that despite this warmth, that the physics of how the atmosphere was processing moisture and then rendering it in precipitation, and from which it subsequently becomes metabolized in the tree, are remarkably comparable to the modern world. And that, in a way, is comforting. That is comforting in the in giving us at least the first indications that we can use some of these warm intervals of the past to make valid predictions for what landscapes might look like in the future as the planet warms. Okay, so we've published some of this. It's all available. This is, we published it in a journal that's freely, open access, and so we have this here. Okay, now let's look at the second type of deposit, which is post-eruptive infills in a kimberlite pipe. And so here we have the giraffe pipe. Don't ask me about the names. There's giraffes, there's wombats, there's pandas. That's nothing to do with me. It's everything to do with the Australian mining company BHP and superstition how it goes is that they only started getting economic grades when they started using weird African and marsupial names. But anyway, we're stuck with giraffe. And the giraffe, and so this is the core that we have. It's a couple of 100 meters long. It was drilled at a 47-degree angle. It bottoms out not in the kimberlite, but in the country rock on the side here. But we do know that the kimberlite itself is Middle Eocene. It's about 6 million years younger than the previous example. And we know that because there was an initial core that did penetrate the kimberlite. The phlogopite was extracted from the kimberlite. It was dated to 48 million years before present. And then the rest of the core was mercilessly discarded, which should be a crime in Australian law. But anyway, that's what happened, and so we don't have that material. We just have the date, which provides the maximum age for the post-eruptive sediment infill. And we have a sediment infill of shales, paper shales, and then we have peat, and in between the two, we have volcanic glass, which we can get further dates on, and which we have, and that volcanic glass dates on fission tracks, which are just radiation damage due to trace radioactive uranium in the glass. We can date, using the fission tracks, the work of John Westgate, we can date these volcanic ashes to 40 million years before present. And that's Lutetian, or Middle Eocene. And that demarcates the lake sediments, and then the infilling of the lake, and then the peat sediments, and then the whole thing capped with glacial sediments on top. What does this stuff look like? One more turn. No? One more turn? Oh, I'm scared if I do too many turns. The stuff looks like, maybe I can just hold this and it'll go faster. No, let me see this. I should have figured this out an hour ago. Oh no! As Francois said, I was weaned working on lake sediments, and I've never seen lake sediments of Middle Eocene age that look anything like this. These look more like Holocene or recent lake sediments that have been, left out in the sun to dry and to bake a little bit. Very, very low density, very, very high organic content, a little bit fissile. So this is the bedding plane, because remember it was cored at an angle of 47 degrees. And so we have things, we call this cookie shale, we call this massive shale, and then there's some places that have paper shales as well. There are regions that are more or less organic, but it's just a pleasure to work with this, largely because most contemporary lake sediments of this age are lithified to some extent. Like if you go to the Okanagan Highlands or the British Columbia extensional terrain for the same period, it's all rocks. I mean, they're nice rocks and they have fossils in them, but they're rocks, which means that you need more aggressive chemical or physical attack to work with them. This stuff is just dehydrated sediment. It is completely unlithified. This white stuff, which was incorrectly diagnosed as bentonite by somebody who works in Alberta, and of course is a little bit biased, and I'm not going to mention any names, but it actually turns out, and bentonite is altered volcanic glass, it's not bentonite, it actually turns out to be microspherical silica, or pure SiO2, amorphous hydrated silica, and we can see with the powder X-ray diffraction, if this was quartz or bentonite or something else, we'd have these spikes. Instead, we see just a pure amorphous silica trace. And so the first thing you think is like, well, it has to be dissolution of silica fossils, dissolution of phytoliths or diatoms or chrysophytes that have been reprecipitated, but we actually see this stuff in close association with siliceous fossils themselves. And so the idea is that presumably during the eruption, water from the mantle was injected into fractures beside the diatreme, and then this water gradually re-entered the lake after the eruption. This water was very, very rich in geothermal silica, and then when it entered the lake at a lower temperature and a lower pH, precipitated out these lovely little microspheres of silica. So this is basically silica gel, kind of like the stuff that you have in your camera bag. And then over 40 million years, there's no evidence of this advancing to more stable phases like cristobalite or tridymite or chalcedony or ultimately quartz. And so no diagenesis, which again fits from the mineralogical perspective with what I'm telling you about the organics. The preservation is simply astounding. And, sorry, this thing is, I need to do 2 back, 1 forward to get it to move. And so in thin section, these are algal cells, diatoms, which is one of my pet organisms, chrysophyte cysts. They have these little resting spores that are also made out of silica. These are the same things that I just showed right there. And then we have green algae belonging to the genus Botryococcus. That should be familiar to anybody who works in petroleum. We can take the petrography and look at these laminations between highly organic and highly silicious, and then look at them under laser scanning confocal microscopy with an excitation and an emission that includes the red part of the electromagnetic spectrum. That's where chlorophylls reflect, and we can see that in these organic zones we have vestiges of fluorescence that are owed to the presence of not chlorophylls and carotenoids, but they're diagenetic vestiges of organic, the diagenetic vestiges of primary photosynthates. We can even get more cute, and this is one of the things that we can do with these kinds of rocks, with these kinds of materials, as opposed to lithified rocks, is that we can extract the pigments just using an organic extract, a little bit of acetone, a little bit of alcohol, and then inject it into a high-performance liquid chromatography system. And this is what biologists use to measure pigments, for example, in water samples or in soil samples and so on. And so this is just the elution time. And now with the current instruments, we can take these individual peaks, and so this is a chlorophyll peak and this is a carotenoid peak, and we can take these peaks and look at their individual fluorescence, which is what we call diode array detection. So this is the wavelength and the fluorescence of the brown peak and the green peak, and we can see characteristic features of these molecular vestiges, including chlorophyll, lutein, zeaxanthin, these are carotenoids, and pyrophyophytin and diadinoxanthin. This is a diatom carotenoid. These are molecular vestiges. It's not perfect. We have some coelution of carotenoids with chlorophylls, but it's not bad either. It's much better than anything that has been hitherto seen from the Middle Eocene, and that's not bragging, that's just the fact. That's what it is. We just got lucky and found it. So the first paper that I published on any of this stuff is now several years old, and it addresses these diatom valves, which are essentially cylinders with a whole cell and then another cell. These are made of silica, and they actually contain the green vestiges of plastids, or of the photosynthetic organs that these microbes use. They're photosynthetic microbes belonging to the group of algae called the ochrophytes. And if you then slice one of these with a microtome and put it under the transmission electron microscope, we can actually see vestiges of membranes and indeed the thylakoids, which is the way these things stacked their photosynthetic organelles before these are primitive to higher plants, of course. Later, the equivalent of this in higher plants is called a plastid, which is simply a package of the photosynthetic organelles. And these are just sheathed in microcrystalline pyrite and are incredibly well preserved. It's just a pleasure to work with these things. Morphologically, If we move to another group of algae, the scaled chrysophytes, that also have silica metabolism but are different from the diatoms because they swim around with a flagellum. So Cynura and Malamonas, these things are still alive today. In fact, in Ontario and Quebec, they're related to things like taste and odor problems in lakes. These are modern examples, and these are the Eocene examples. It's the first report of this whole group of organisms from the Paleogene at all. And so we're entering a little bit terra incognita. Admittedly, they're only unicellular photosynthetic microbes, but they nonetheless tell us quite a bit about the evolutionary history of these important algal lineages through time. And so where we are with this lake sediments and what we've surmised so far from essentially the stamp collecting aspect of documenting these microfossils. So this is Botryococcus. These are called birotules or spicules of sponges, freshwater sponges, which typically live in very acid, high dissolved organic carbon lakes. These are chrysophytes. These are pennate and centric diatoms. This is that beer can shaped diatom that I showed with just the plastids. These are scales of testate protozoans, rhizarians, if you'd rather. They're small amoeboid organisms, and these are pollen grains. All of these are exceptionally preserved, and without exception, each of them has direct affinities with modern organismal groups, and we can put them not only to families or broad groupings like pennate diatoms. We can say that this is a eunocioid diatom belonging to the genus Eunocia, and that its closest relatives today live in low pH, low alkalinity, high dissolved organic carbon lakes. We can make these paleoecological inferences, and so morphologically and ecologically, these things all have very, very, very readily identifiable modern extant relatives, and we would then say that at least for the last 40 million years, for these groups, for which many of them have a pretty patchy fossil record or no fossil record at all, including the scaled chrysophytes, that are right now we're standing our ground for this notion of protracted evolutionary stasis, which is saying that these things don't appear to have evolved as much as we perhaps thought they might have. They don't appear to have evolved very much at all in the last 40 million years. You can use this information, and I have been, and this is another example of how this is moving in all sorts of new directions, is that I've taken a liking to a few people who are molecular phylogeneticists. And the only molecular people I really like are the molecular people who don't just see the organism as a vestige of DNA in a microcentrifuge tube, but they're actually also interested in what the organism truly is. And so there's really two types of molecular people. And the group that I'm working with, in particular a guy called Debashish Bhattacharya at Rutgers University, what they have done is they've compiled from, oh no, what happened there? They've compiled from modern scaled chrysophytes a database of nuclear, mitochondrial, and plastid genetic markers. So these are all relatively conserved. Of course, because of when these algae evolved, there's a lot of horizontal gene transfer. They don't only evolve from descent. For example, the plastid was an organelle that was acquired by endosymbiosis. And so you do need to have the independent markers from different parts of the organism, which fortunately these geneticists are clever enough to do. And then we can take our organisms from the Eocene and place disciplining mileposts on the evolution for which the topology is given by the genes. We can then discipline this with the fossils from the Eocene. And we'd do it with other fossils if we had them. We just don't have them yet. And what this ends up producing in this group of organisms, much as it does in several other but not all lineages, is that these things originated much, much earlier than is presently understood by the fossil record, and that ghost lineages, which are periods of time ranging from tens to hundreds of millions of years for which no fossil record is known, are ubiquitous features of the phylogenetic or the evolutionary history of these organisms. And so it did look at one point, so we've done this now with the chrysophytes, we've done it also with the freshwater diatoms. It did look for a long time that in terms of algal evolution, that the diatoms and the chrysophytes, these siliceous algae, arise very, very recently, perhaps in the Miocene, and then radiate tremendously. The diatoms now dominate ocean photosynthesis, about 1/2 of ocean photosynthesis, which for a single group is huge, that they had fundamentally different evolutionary patterns compared to other algal groups, like green algae, red algae, seaweeds, and stuff, which we know go all the way back to the Proterozoic. And now, and that discrepancy is a little bit unnatural, because after all, they're all just like tiny little green slimy things that photosynthesize. It actually now turns out that some of this evidence is supporting at least a unified view for the ancestry of unicellular algae. And I know that's a long way from dinosaurs in the Badlands, but nonetheless, it's pretty cool. François, how am I doing for time? How long do I have to go? Because I didn't think it was going to be this late. It's an hour talk. Are people, because I can end here, I can make a few final statements. And what I'm going to do is oh my goodness, am I really? I'm sorry, I'm slow today. Okay, so we're done with the lake, we're done with the ancient wood. We're going to do one last thing, and it's perhaps the most important. We're going to go into this peat where we have terrestrial vegetation. As I said, the lower part of this peat is bounded by this volcanic ash, and so it's really lovely. And at first we thought it might be kimberlitic, but it turns out that the chemistry, so these are the vesicles in the volcanic ash and electron microscopy, it's 40 million years old, so this is the lower bounding for this peat when the forest colonized the lake having been infilled by sediments 5 or 6 million years after its formation by the kimberlitic intrusion. It's just such an amazing story. It actually turns out that this ash, based on this chemistry, probably came from Alaska, or probably from the Alaskan peninsula, Kamchatka to be precise. We can actually fingerprint the geochemistry of these ashes pretty well. Here's a chunk of wood. It's metasequoia, same as you saw before, so I won't show you any more xylotomy. That's bark, and this is the 47-degree angle of the core. The core just went right through a piece of wood, and the wood is unpermineralized. Some of the peat has seeds. This is a wingless pinaceous seed. This is in my freezer. It's the one we're going to sequence DNA from. And we also have amber. So for paleobotanists, how could it get any better? I mean, I'm so happy, which is why I'm allowing myself to go over time before lunch. But I am starving, so I'm gonna hurry up a little bit. Here's some of the taxodioid foliage, and so it's beautifully opposite leaves. This is unpermineralized metasequoia. Under the electron microscope, it looks kind of blurry to me, but hopefully it looks better to you. We can zero in on an individual leaf, and we see, so this is the central axis, 2 rows of stomata, which are the gas exchange vacuoles. So this is where all the action takes place in terms of water vapor and CO2 entering, and then it gets processed and sent to Rubisco, where the dirty work of photosynthesis takes place. And so we have, and again, if you were to take this and compare it with a modern metasequoia leaf, say from the Muttart Conservancy, but a conservatory in Edmonton, but who wants to spend $9 to go in and it's total ripoff. But if you were to do that and steal one of their metasequoia leaves and not get busted and put it under electron microscope, it would not look any different from this. In fact, it would probably have leaf wax cuticles and pesticides and stuff on it that it probably wouldn't look as good as this. We've done that comparison. We can also do the Tritzi autofluorescence. So this is the same trick I showed you with the shales, and we see vestiges of of photosynthase, of chlorophylls in the red part of the electromagnetic spectrum in here. And so we not only have morphological, but we also have geochemical preservation. And this is an even more tantalizing one, where the leaf has been stained with ethidium bromide, which typically sorbs only to nucleic acid. So this is giving us a positive response in this very narrow band of fluorescence for the presence of either RNA or DNA. I think this is a false positive. I don't think there's any DNA present in these 40-million-year-old fossils for many, many reasons. A, I'm going to get put in a nut house, but B, because I think the hydrolysis, the hydrolytic degradation, and the cross-linkages of DNA preclude preservation at this time scale. The oldest DNA that we know of that's reliable is about 120,000 years before present, and that's in the bottom of Greenland ice cores. I think that this fluorescence is actually a false positive for structural proteins, for example, heat shock proteins or legumins or other plant proteins, and we're starting to work on that a little bit with my friends from biochemistry. So I'm just quickly going to run through what we can do with this peat record, this fantastic paleobotanical record, and bring in essentially the reason. So up and now it's been mostly my work and my thinking, and why I've brought on people like Dana Royer and Dave Greenwood and Art Sweet to look and to process this record. And, you know, what does this tell us about climate? And what does the reconstruction of climate, including the composition of the atmosphere, mean for the future? And I think we're getting to the point where we can now use this record, because of the quality of this record, to say, okay, at X concentration of atmospheric CO2, the equilibrium climate response is this, and how does that compare to the trajectory that the planet is currently on in 2013? And so we have pollen grains. This is Art Sweet's counts. They're preliminary counts, but they're good enough for our business. They're dominated by the cupressoid conifers, a few pinaceous things. Beautiful representation by the Phagalian trees, so the beeches and oaks, and this spectrum, so with chestnut and hazel, and I don't want to say anything that might offend Dennis here, because I'm not a paleontologist. I spent 2 years in a paleontology lab when I was a kid. These is sort of warm temperature, sort of Tennessee-ish, Alabama-ish, although... Yeah, so it's definitely very, very different from today's modern pollen spectrum. David Greenwood has adapted a technique called a transfer function, where you can take the pollen frequencies using a coexistence analysis with the ecological tolerances and optima of the closest modern plant taxa, and then you apply that to their Eocene counterparts, and you back out temperature. And when you do that for this sequence, from the lake sediment into the peat, let's focus on the peat here. The coldest temperature is never below 0 degrees Celsius, and that kind of matches with what we know from the ecology of Metasequoia. And the mean annual temperatures are somewhere between 12 and 17 degrees present, and so the temperature is about 17 to 22 degrees warmer than present, and the coldest months, for which the coldest months are somewhere, you know, between 0 and 6 degrees Celsius, with a mean from the peat of about 5 degrees Celsius. That is fundamentally different from today, where, of course, as we know, the coldest months in that part of the Northwest Territories is in the range of -20. So then, what was the ambient carbon dioxide concentration to produce this approximate or estimated climate response? We can then interrogate this by the density of the stomata, those are those gas exchange vacuoles, because the plant will make fewer of these highly specialized cells if the partial pressure of CO2 is high enough for its metabolic engine to run without needing to accrue added CO2. And so we have an inverse relationship between the density of stomata and atmospheric CO2. So these are little metasequoia saplings that we grew in a controlled growth chamber in Connecticut. And then it's really good with metasequoia because the needles only last 1 year. So you let it equilibrate for a year, and then it grows for another year, and then another year. And so it's the length of a graduate degree. And then you clean them and count them, and you get a result like this. Of course, the proxy, so the idea is then to use this relationship and then bring it from the modern ecological space to the fossil ecological space and reconstruct atmospheric CO2. It works pretty well. So the stomatal index is simply the density of stomatal cells to total epithelial cells per unit area of leaf. It works really, really well for this sort of low zone. So today we're at 394, that's right here. And these low ones are actually from museum collections where we know the date of collection, we know the corresponding atmospheric CO2 from the ice core record. So there's lots of clever historical business into this, and that's why we work with people at Yale, because they have the Peabody Museum that has specimens going back to the discovery of this plant in the 40s, but we do end up with a situation where the proxy gets saturated above a certain CO2 concentration. The tree just can't be bothered to make fewer stomata, is basically how the tree feels. And I don't want to sound like I'm anthropomorphizing a metasequoia tree, although of course I kind of am because it's my favorite thing in the world. So stomatal indices, this doesn't look like a lot of work, but it's a master's thesis. And so each of these is about a dozen different leaves and the measurements. And I'm just going to stick to, and I want to finish up here. I want to stick to the key points. What we end up with are a range of reconstructions between about 450 and 900 parts per million. So higher than today, but not thousands of parts per million. Hundreds, well within the range of atmospheric CO2 that humanity, with known petroleum reserves, known economic forces, and barring nuclear explosions or geopolitical instability, well within the range that we'll be able to put our planet within a matter of decades, not centuries. Of course, we can't really say much about these values because of the saturation of the proxy, but tantalizingly close to modern values up here at the top of the peat. So let's just take the average of about 715 or so parts per million for a long-term equilibrium climate response that then produces winters with no frost, precipitation at least 4 times what they are at present, and mean annual temperatures in the 10 to 20 degree range. So compared to other reconstruct, and there's a real black art to reconstructing CO2, and I'm going to say the stomatal indices are no different, but at least they have a direct ecophysiological linkages to how plants perform. But there are all sorts of other ways involving the boron isotopes, the C13 of biomarkers, each with their strengths and weaknesses. And typically, they give a slightly higher range of responses for the Eocene, in many cases above 1,000 parts per million. And so today, we're down here, and we've been down here since at least the early Miocene, a little bit of a transient in the mid-Miocene, and then the greenhouse, ice-free world of the Paleogene, and so the Oligocene, Eocene, and Paleocene, and then the Cretaceous is back here. We're somewhere between 500 and 1,000, and our results would suggest somewhere more like 700 on average, and that is well within where the planet is going to be almost certainly. I don't want to proselytize and so on, but there is a reason to put this in the contemporary context. A, because I'm concerned as a father and as a human about climate futures, and B, it's pretty interesting scientifically, and C, I think we can actually contribute something to this in a meaningful way. So if we take these as a fancy way of saying the average distribution, so like probability, density functions, that's just what us scientists do. We use jargon, we use $10 words when we can get by with monosyllabics. But anyway, here we have the 714 plus or minus 180 parts per million for the middle Eocene atmosphere. Not a lot of power, but you know, it's hundreds, not thousands of parts per million. It's the kind of atmosphere that will certainly exist in my child, who's 6 years old, in his lifetime. The pollen would suggest that the equilibrium response of the temperature was about 15 degrees of mean annual temperature, so about 20 degrees warmer than present for this latitude of the North American continent, and that delta T is even greater in the winter because we had winters in the Eocene that did not have frost, in great difference to today. We can take this is simply the position of the empirically derived mean annual temperatures from the cellulose of the metasequoia wood, not from the panda that I showed for 53 million, but for the equivalent wood in giraffe. And they're just different solutions depending on how you tweak relative humidity. And so you can forget about this, but essentially if you adjust the relative humidity to be somewhere in between 64 and 77% relative humidity, you end up with the same solution, and something like this, 83% humidity, probably a little bit too high. So most of our proxies are converging. And this is sort of my synthesis slide, and this is where this is going in my thinking, and this is my current thinking of how we can take all this paleobotanical data and perhaps make it relevant for the future. And so we have something called the Intergovernmental Panel of Climate Change, which has some strong points and a lot of negative points, who predict that for a doubling of atmospheric CO2 from the pre-industrial level, which is about 280 parts per million, so 290 to 560, the solution is a climate that's about 3 degrees warmer, and so this is delta T relative to the pre-industrial value. Since the pre-industrial time, we've already had about 1 degree of warming, about 0.8, so that's where we are at present, and that's what we call this Charney sensitivity, in deference to the departed British meteorologist Jules Charney, who actually came up with this number of 3 degrees plus or minus 1.5 for a doubling of CO2. He came up with it in the 70s, and the IPCC has not been able to improve upon this. We call this climate sensitivity. It's the response of temperature to an increase in CO2, and CO2 is, of course, acts first and foremost as a radiatively active gas in retaining outgoing infrared radiation, and these are essentially what we call the fast feedbacks. This is what happens instantly through the physics of infrared radiation retention and the changes in the Earth's reflectivity, in particular the waning of snow and ice cover, and making the Earth more absorbent versus more reflective. And these we call fast feedbacks. In the Eocene, it's different. These are what we call the slow feedbacks. And this incorporates the progressive warming of the global ocean and the processes that take about 100 to 1,000 years to stabilize. And we often hear the term that, oh, we're at present and we've had a little bit of warming, but oh, there's all this warming in the pipeline. We hear that a lot. The warming in the pipeline, of course, is real. And this is once the slow feedbacks, for example, the long-term response of ice sheets, the warming of the global ocean, changes in oceanic circulation. When these gradual processes kick in, as a response to the fast feedbacks, then you end up with a temperature response which is much greater. And this is what it's been estimated globally. And so the combination of the slow feedbacks and the fast feedbacks has another fancy name called Earth System Sensitivity. And there was a paper in Nature just a few weeks ago, and this is the best estimate from the paleo record from the Pliocene, about 3 to 5 million years before present, where CO2 was somewhere around 420 parts per million, and the climate response was somewhere around 3.5 degrees warmer than present. And we can see that as CO2 increases, the amount of slow feedback or slow inertia in the climate system only increases. So what about our proxy data? Well, there's a few interesting points. The first is our Giraffe Middle Eocene, which is 64 degrees latitude, is up here. And then for the Pliocene, with this relatively modest CO2, only slightly greater than today, we have a fossil record from a place called Strathcona Fjord on Ellesmere Island, about 80 degrees latitude north, that from similar proxies to the ones that I've just shown, except not the stomatal indices, but the biomarkers and the isotopes and the pollen and the wood would suggest that the temperature was somewhere around 20 degrees warmer than the pre-industrial average for the same part of the world. And as I've just shown, we have a similar amount of increased warming for the Giraffe site. And the difference between the equilibrium long-term climate response, which is, of course, the one that's reflected by our proxy records. The difference between this and this is this phenomenon called polar amplification, by which we get greater relative warming or cooling at the high latitudes relative to the global average or the tropical latitudes. The interesting thing is that this polar amplification is sustained even as the threshold is crossed between a world with an active cryosphere, that's sea ice and glaciers that actively contribute to energy balance by essentially being highly reflective, from the world that largely lacks a cryosphere. And the view is that this threshold occurs somewhere around 500 parts per million. So we're at 394 right now with 2%, 2 parts per million per year on average. So this is somewhere around 50 years from the future, we'll be in a situation where the physics of the climate system are no longer able to sustain continental ice sheets and sea ice, and that will engender, of course, the transit from the fast feedback into the slow feedback world that portends really a world that even at the high latitudes and globally is much, much warmer than present. So for the subarctic latitudes, this Charney sensitivity, only the fast feedbacks, is, chicken scratch. It's a minor component relative to the slow feedbacks. And we also, even in a world in the Eocene where we think that sea ice and glacier ice were at a real minimum, we still have the observation of this polar amplification. And so this persists even in the absence of planetary cryosphere. That's like what scientists say for snow and ice. I'm sorry about that. It's just the way I'm wired. In the absence of sea ice and glaciers, we then have to invoke other mechanisms for keeping the high latitudes extra warm. And I think that heat storage within the Arctic Ocean, particularly as the Arctic Ocean becomes ice-free, is really, really important. And there's other work suggesting that water vapor and things like isoprene, which is a plant volatile emission, and its feedbacks with aerosols and cloud formation may also be highly relevant. The one thing is that where we're going as a planet, everything from the paleo record, and of course the paleo record does not speak to the political timescale, the human timescale, the 4-year timescale, or whatever. It speaks to the equilibrium solutions that include the slow feedbacks, but even at a relatively modest 500 parts per million, it portends a fundamentally different world than those of present. And and that means that, well, 3.5 millimeters of annual sea level rise is not going to decelerate. That will accelerate, and it portends a really, really different world for the future. Maybe not in my lifetime, but certainly that of my kids. My colleagues in Germany, Jörg Pross, and then other people like Greenwood shows up here again, and a whole bunch of cast of characters have verified that polar amplification in the Eocene is quite true with this beautiful science paper from Seymour Island, which is basically antipodal to our site in the Northwest Territories in the Southern Ocean. So this is sub-Antarctica during the Eocene, and lovely, lovely fossils and palaeontology that are characteristic of modern settings in Australia, New Guinea, and New Caledonia in the subarctic latitudes of Antarctica, including palms, and they have some lovely, lovely fossils. I'll conclude by saying that if we take all this information from the fossil record and then fast forward it to, say, 2013, what we're seeing and what's going on the planet today, and so most people realize, or most people have heard, that 2012 was a record low sea ice in the northern hemisphere Arctic, so dipping below 4 million square kilometers. No one thought that 2007 would be beaten, which was the previous record in a very, very warm summer. The summer wasn't particularly warm this year, but it still beat it, and by a fair margin. So this is the long-term average from 1979 based on satellite-driven remote sensing imagery. So the sea ice reaches its absolute maximum. And this is essentially a proxy for the area of high albedo, of highly reflective surface in the high latitudes of North America. Well, what else happened on this summer? A strong positive feedback, because air temperatures weren't particularly warm this year. Some other dynamic, and most likely ocean temperature, were related for this exceptional loss of sea ice. And now it looks like at this rate, Arctic sea ice might not even be a feature of the planet in summer 10, 15, or 20 years from now. In August last summer, there was a development of this extremely low-pressure cyclonic system. Typically in the Arctic, cyclones are derived from the North Atlantic or the North Pacific, and then they come up Baffin Bay or through the Bering Strait, and then play out in the Arctic. Having storms that are born from the Arctic Ocean, as the Arctic Ocean loses ice, and the heat of the Arctic Ocean, keeping in mind that water has 4 times the heat capacity than land, and that this heat is no longer kept from the atmosphere by a blanket of ice, the active heat exchange between the ocean atmosphere appears, at least in this case, and it is, of course, an individual example, leads to a situation where the Arctic Ocean, in and of itself, can create significant cyclones. This is just like a weather geek map. This is what it looks like from the satellite. This is a meaningful storm, which then had, of course, a very, very powerful positive feedback in terms of mopping up what remaining sea ice was lying around some of the Arctic islands. I don't know if you can really see what's on here. That's Ellesmere, Baffin, Melville. So that's the Queen Elizabeth Islands. That's Beringia and Siberia and Alaska over here. So that's where it's sitting. And it formed on the Arctic Ocean, was created in the Arctic Ocean, and died in the Arctic Ocean. And I think this is just a foreshadowing of the synoptic processes that are required to explain how in the Eocene in the Northwest Territories, precipitation was 4 times what it is at present. Well, today, in Yellowknife, precipitation comes only from the Pacific because the Arctic Ocean doesn't do much. You ramp the Arctic Ocean up, you enable that precipitation to be deployed in the circumferential regions, and then all of a sudden it is wet, and because of the equilibrium response to a slightly higher than present CO2, it's also warmer, and then there you have your subarctic metasequoia forest. And so just to summarize, hopefully I've demonstrated that kimberlites are useful for something other than diamonds and mantle petrology, although those, of course, are noble pursuits in and of themselves. That the Middle Eocene climate was quite a bit warmer than today, but also has components similar to today, which enable us, as long as we're judicious and careful, to apply principles from modern meteorology and modern botany and a few other fields like counting stomata in a dark room in Connecticut, to make this all fit together in a story that will inform us not only about the past, but also of the future of the high latitudes. And so we're working on a bunch of other stuff. I have no idea what the BHP's sale to Harry Winston means for us, but we're also working on older deposits. Misery seems really cool because one branch of the Misery pipe is actually been redated because all the phlogopites were Archean in age, so they date the country rock, which means they're detrital, and now we have perovskite, which is a titanium mineral, and the perovskites are dating Cretaceous. So we might even have unpermineralized wood of Cretaceous age in one of these pipes, and myself and my students in our group, we're just going to continue on with this, whether BHP is behind us or not. And then the final thing is that, if you have a little kid like I do, and this isn't a real picture, it's the biosphere in Quebec. If you have a real kid and you really want to take your kid paddling on the Arctic Ocean with an active cryosphere, you should do it fast. And all my stuff is available. I break every copyright law in the world. If you're interested in any of the papers, they're available. And I also have further comments on, what I think the nature of some of these phenomena, both physical and socioeconomic and geopolitical, some of these phenomena involved in both present and ancient climate change. I also have comments on my website as well. And so I'll leave you here. This is Western Svalbard and Spitsbergen. I'm sorry for going so far over time, but I really did. So let's see what I get after lunch.